we've given the prize right now, but I think we should also congratulate FreePad for organizing the meeting so greatly. And also, I mean, India, you just like, the hospital is so great. So FreePad, I don't know where you are. I think he's not here. I've got to do it again. So well, when he comes back, I'll clap my hand and just like, you'll do the same. Oh well. So back to work. So what I would like to talk to you today is some kind of a new mechanism of action or new properties of metronomic therapy or chemotherapy based on a very simple model. And actually what we did is just like introduce the concept of tumoral heterogeneity in metronomics, and we wanted to really go back to basics. So it's going to be a pretty simple thing. But before I start for good, I really hesitated because we have 3 projects that are pretty mature now around metronomic preclinical data. So there's one that is really interesting that is based on the association between AKT inhibitor and metronomic, and it gives also new insights through studying metabolisms of cancer cells and how they could match together well. And there's another project that is also pretty much done now, which is how we can use mathematical modeling that Sebastian told you about yesterday to optimize the dosing of metronomic chemotherapy. And so this is a project regarding metronomic gemcitabine in neuroblastoma, and it was pretty successful. But this is going to be for later. So Eddie told you about heroes. So I also have some heroes. We have some in common. And so when I was a teenager, I discovered evolution and Charles Darwin, and it was really a passion for me. So this is the old Charles Darwin, and this is the young Charles Darwin. And you know, look at him well. I think it's important for the future. So basically, as you all know, Charles Darwin just like published that great book called The Evolution of Species in 1859, and well, he basically described for the first time that, you know, any living creature was evoluting, all the species were going evolution, because basically you get like different small differences in all individual in a species, and this difference make that you can compete, you can reproduce better, and so if you can reproduce better, this small difference will get like dominant in the population, and then slowly it transformed the whole species. And in 1976, Noel published that paper in Science, and he was the very first one to introduce evolution in oncology, starting from a normal cells to its transformation into a cancer cells into an aggressive cancer that would be eventually resistant to treatment. And the evolution in cancer is now something big again, really, really big. And it was put back at the center stage 4 years ago now with the team of Swanton. And they published in the New England Journal of Medicine how, and they were able to demonstrate actually based on genetic mutation, how just like this cancer clone evaluates in the body, within the tumor and within the metastasis. So this is this very paper. So they just sampled the tumor at different sites, sampled different metastases, and they were really able to draw the branched evolution of a given tumor in individual, in several individuals though. So this is very, very interesting, of course, as a concept, but it's also very interesting for the patient. And we know that in a tumor, all these different cell clones are present in the beginning, very early in the beginning of the genesis of the tumor. And this is one of the reasons why target therapy may work and then stop working. Because this is a very famous case report that has been published in a GCO 5 years ago. So this is a patient with a metastatic melanoma, obviously heavily metastatic. And it was treated by a BRAF inhibitor. And you had, after 2 months, a beautiful response, incredible response. But 2 months later, the tumor started growing again. So they were able to demonstrate that the clones actually had another mutation in MEK1 that was responsible for the resistance to BRAF inhibitor. So, starting from this, what can we do? So, I have my own heroes. So, I told you about when I was a teenager and I discovered Charles Darwin. This is when I was younger, and I'm also obviously a Star Wars fan. And when I started doing research, I had another hero with its Robert Kerbel. And if you look at them, well, don't you think it's funny, isn't it? They look like I don't know how you call this, rouflaquette, we call them in French, but I guess either if you just like cut the rouflaquette of Darwin, or if you have your rouflaquette grow, you would just look pretty much the same. So I don't know if it's your good profile, but well, so the only way I found to just like to have my 3 heroes together was to go for the Clone Wars. So basically, Clone Wars, that's the Force, that's metronomic, just like Bob, and Darwin is here for evolution. So, there we go. So, actually we had the idea after the very last day of the metronomic meeting in Marseille. So I was quite relieved because the meeting went on well, and so we were with Eddie, and maybe drunk slightly too much wine, good wine, and so we came up with a concept at the time. So the project is being led by Manon Carré, who is a senior researcher in our lab, and Marina Barandenko is the PhD student, and then the two of them are doing a great job on this project. So the first thing was really a basic question, right? Because this fancy genetics data, but our first step was like, how do these clones interact with each other, or do they interact with each other? But it's striking because when you start with a tumor, you know that the resistant clones are inside, but they just like the tumor sensitive, so they just like kept to a minimum to some extent. So we said, well, I guess to some extent these resistant clones, they have to be inhibited in some way by the sensitive one. So that's the first step. And then if there's indeed an interaction between these clones, there's this impact on the way we should treat them and use our chemotherapy. So we focused on MTD and obviously metronomic. So we needed a model, so very simple model. We used a non-small cell cancer, lung cancer cell line, very available, and we have a resistant derivative. So this cell line is resistant to 40 nanomolar epithelium, which is an antitubulin agent. And so we fluorochrome them in red and green. So that's a nightmare for me because I'm totally colorblind. Right? So you have to have compassion from now on, because for me it's just like not green or red or red, right? So we have the sensitive one fluorochrome in red, and we have the resistant one fluorochrome in green. And so the thing is that we can easily follow them when they grow together. And we also generated like, you know, the different combinations to have some control. So as you can see here, you have the IC50s regarding some major anti-cancer agent, and so you can see the resistant one are not only resistant to epithelium, but also to paclitaxel and to a lesser extent to cisplatinum. Then we needed a device to explore interaction between the two clones. So we have that little device with an insert that separates the seeding zone in two. So you can have the resistant one inside and the sensitive one outside, or vice versa. And when they start growing, you take off the insert and you can watch how they just like interact with each other. And so we can measure the fluorescence globally, so you can measure and evaluate the evolution of the global population, but you can also use the microscope, and then you can identify some cell that may be remaining at some within the global tumor volume or tumor cells. And then we generate a second model to be like slightly better than just two-dimension model. So we have spheroids that have the two clones. So we have two kinds of spheroids. We have the mixed one, where it's just like a mixture of sensitive resistance, and you have the second model where you have a layer of sensitive outside and a core resistance, or here again vice versa. And the good thing is that the number of cells that you have is really nicely correlated with the fluorescence. So we can measure the quantity of a given clone within the tumor and eventually locate it within the tumor. Oops. So first we just like see what happens. So as you can see here, if you let them grow together, you got the sensitive clones that keep on growing, and you got an inhibition of the resistant ones. And as you can see more precisely on the microscope pictures, they just not only not grow, but they disappear. So the growth is inhibited, and then they also die. So that was not at all what we expected, but that was a good news because they were interacting. And so we got some quantification of these experiments here. So the difference were significant. And the important thing is that when we tested different cell lines. So we also had this VP16 resistant cell line, unconscious cell line, and we just like find the same thing. And we tested also a colon cancer cell line with that was resistant to another agent, here oxaloplatinum, and we found the same phenomenon. It was not as strong, but still we had the same process. So to be true, in some cases the clones don't interact with each other, and in some clones it would be the opposite. So the resistant clone can inhibit the sensitive one. So it's not like a universal concept, but it was. I think we thought it was worth going forward. So then we went to see what happens when you have these three-dimension co-cultures. So we have two kind of spheroids. We have the B-colored homospheroids. So this one have at the same time the resistant green and the resistant red. So we have some kind of control. And we have the B-color heterospheroids who have the green sensitive and the resistant red. And so if you look here, we quantified the number of resistant cell, red, in the spheroids, and you got here on the top the homospheroids, and on the bottom the heterospheroids. I mean in full line. So we basically find the same results, right? If you got the homospheroids, so just the resistance, then nothing will happen. But if you got the heterospheroids, then the number of resistant clones within the spheroids they don't increase. So you have the same process that the sensitive clones can keep growing and they inhibit and eventually kill the sensitive one. So next question was, how does it work? Is it some kind of direct inhibition or distant inhibition? So we designed that simple experiments. We had the resistant on the top, sensitive on the bottom, and then a membrane, macropore membrane that separate the two cell lines, and so that no cells can just like get directly in touch. And if you look at this, the bottom panel, you see that the sensitive clones, so this on the top, they keep growing over time. And if you look at the resistant one, initially for 3 days they grow and then they stop growing and they just die again. So it means that basically you don't need the direct interaction to obtain this phenomenon. So here again, to strengthen this finding, we just decided to measure the evolution of the number of cells. So using this experiment, and we just expose the sensitive or resistant clones to the sensitive supernatant medium in which the cell will culture. So if you expose the resistant cells to the sensitive medium or the resistant medium, you see that you got for 48 hours, you see that you reproduce the inhibition of growth. So it's another argument to say that this is really something that is in the medium, the supernatant. And this was statistically significant here again. So we wanted to be slightly more precise, so we separated the extracellular vesicles from the supernatant and tested the whole supernatant that had the 33% inhibition. The extracellular vesicle didn't do anything, and the subgroup fraction of the supernatant just was able to do just pretty much the same as the total supernatant. So it means that it's the inhibition of the resistant clones by the sensitive clone is at least in part mediated by a soluble factor that is present in the supernatant. So we went we have more data about how it works, and we get a pretty good idea now. But since I don't have enough time, we're now going to move forward to what happens when you use treatment. So you've seen that slide like over and over for the last 2 days, but I think it's worth having a look at it again. So we think that metronomic, or we thought that metronomic is initially mainly anti-angiogenic or anti-vascular, and so it's getting more interest because of all the checkpoint inhibitors that it may also act, or it acts on the immune system. But remember, it also acts on cancer cells. And Bob said yesterday and the day before that we should reevaluate how metronomic therapy acts on the vessels. But I think we should also reevaluate and be aware that metronomic can also directly kill cancer cells. And this is one of the reasons why we think we should be re-evaluating it. That's the little Indian cancer cells. You know? So So basically same models, but then we used MTD chemo. So doses of epotylan that would be like IC50 to IC90. And so this is what happened, right? You start treating. So initially the sensitive cells start growing and then they get killed by the chemotherapy since they are sensitive cells. And as they die, the break that the inhibition of the growth of resistant cells is lost, and you end up having no sensitive cell lines but only resistant clones. So in this two-dimensional model, giving metronomic if you have resistant cells is not going to work, and you end up with having only resistant clones. If you do the same experiments with the spheroids that we talked about, so the B-colored heterospheroids or the B-colored the B-colored, heterospheroids and non-treated or MTD treated, so you see at D0 and D6. So you see the color basically is changing, right, and it's getting all red. Tell me if I'm right. And if you measure what's going on, you see that in the MTD treated spheroids, you end up having basically only resistant clones. So the same process occurs in this three-dimension co-cultures. Well, if you use metronomic, different things happen. So, and this is also why dose matters. If you use, so this is the control on the top, right? So if you use like no dose, of course, you got your sensitive clones that keep going. So you eventually don't have or have very, very little resistant clones, but overall your tumor keep growing. So it's not working. If you use a small dose of metronomic chemotherapy, you got an inhibition of the sensitive cells. You maintain the inhibition of the resistant one, but in the end of the day, it's still a growing tumor. Well, if you have a bigger dose of metronomic, then you maintain to have at the same time an inhibition of the growth of sensitive cells, but maintain, because you still have enough sensitive cells, but then maintain the inhibition of the resistant ones. So, and if you use like metronomic doses that are too high, then you kill the sensitive one, and you end up with having just like the resistant cells. So metronomic may be a way to at the same time control the sensitive one, but maintain the inhibition, but you got to find the right dose. So, and if you do the same experiment in a Transwell experiments to see if the inhibition, the mediated inhibition still works, this is what happened, right? So if you treat with, if you don't treat, that you see that you got an increasing number of sensitive cells, but also a fair high number of resistant clones. If you use the high dose the high dose chemotherapy, then you kill all the sensitive cells, and you end up having like a lot of resistant clones. And if you use the metronomic, you see that you decrease the number of sensitive cells and limit the proliferation of resistant ones. So clearly here again, even in the like separated model, you still have a better result if you use the metronomic dosing. And if you use the spheroids model, well, you see that you get pretty much the same results. So if you don't treat, you got an increase in the number, in the volume of your spheroids. If you use the MTD dosing, then you get a slight decrease in number, but that you got only resistant clone. And if you look at day 20, then you have like a very much bigger tumor because it's just made of resistant clones. And if you use metronomic, you see that you manage to have a control of the tumor, and intrinsically, as you see on the bottom, it's made of both resistant and sensitive clones. So I'm getting close to the end. So there's also some point that came out of the model that are really interesting, because in the area where the resistant clones were, not all the cells die. You have these cells that you look like these fat big cells, and if you use the high dose chemo and kill all the sensitive clone, they start regrowing just like normal. And if you test all the anti-cancer agents, they just behave exactly like the resistant one. So we're working on to know what's going on in these cells and what exactly what state they are in. So basically, with this very, very simple model, basic things to understand how clones can interact, we were able to demonstrate that clones interaction can impact on their growth, on their survival, that this interaction is at least important. This model mediated by a soluble factor. Of course, we need in vivo validation, because this is just like spheroids, so we have to see what happens in animal at least. But we have to integrate this finding and these potential findings in the way we give chemotherapy at least, but I think it should be really applicable to target therapy, and we're working on this right now. And so the good thing with metronomic chemotherapy is that it avoids the selection of resistant clones, it limits the proliferation of sensitive clones, and globally it can preserve the inhibition of the resistant by these sensitive clones, and globally you maintain a biodiversity within the tumor. So I would like just to, we just talked about the Metronomic Global Health Initiative. So just to tell you, you're all welcome to join in. And we're gonna have another Metronomic meeting at the end of May, because they wanted to organize it on this very same year, on the very same month, for the people living in Central and South America. And we have a very interesting program. So if it's not too late to join in, and you can have a look at the program, because we're gonna have like really exciting data that will be presented there too. And I would like to thank all the people I'm working with around Metronomics, and all the donors and grants that we were able to get over the last years. Thank you for your attention.